Thank you, and thanks, Yue, for inviting me to come to this talk. So today I will be mainly talk about my packages, the lazy slide, which is basically Python packages for analyzing the whole slide imaging. So this is a package that's jointly efforts by me and a PhD student in my lab, Abila Ernesto, and uh, my supervisor, Andrew Rendero. So let's get started. So this is a example of host line images. As we can see, the nature of the human tissues are quite contains information from multi-scale. So if we look at this image from a very high level, you can already see some granularity, some structural differences at different part of the tissue. But if we zoom in, you can see even more details. You can start to see some uh, curvature, some different part, like some muscles, some um, uh, part of the tissues. If we even zoom in, you can already capture the single cell level. You can already see the nuclear stairs. So this basically tells us that the whole slide images contains a lot of informations that's probably not that's like underestimated by our previous research that we can probably covers much more than we thought from these images. So the digital slides, this whole slide image data sets is actually a gold mine from our perspectives because it's first not widely used in scientific research, like not really dig up. And compared to spatial omics, it's much cheaper to generate, and you can generate much more host-like images. And I'm not saying spatial omics is not good. You should use it as long as you have the money and the technologies. But host-like imaging can be a very nice way to approach a lot of biological questions in a much more cheaper and quantitative way. So also given another consideration is that we are at the eras of many foundation models. So for example, some general self-supervised vision models, such as the recent release of the Dino V3 from Meta. And for example, the CLIP model that combine both the text and vision from the OpenAI, and obviously the ChatGPT and many other LLM model or VLM model we use in our daily lives. All of these foundation models basically give us a way to analyze this all kinds of modalities in a very cheap way. And if we try to transfer this model with pathology data, so we can try to convert them into, let's say, pathology-specific foundation models. So for example, Mahout Lab from Harvard, they developed different kinds of foundation models, such as UNIT, which is a vision foundation model, and Conch, which allow you to use both text and images to conduct analysis. And with all these tools in mind, we can actually do many things with these host line images. But before I go details into what we can do, let's first take a look at like what exactly is the host line images, how does it exist in our computers, how do we store it? So because as you know, after we scan the host, the tissue slide with whole slide scanners, um, it's actually very big. So it is almost impossible to read these whole images into your memory, into the computer's memory. For example, this slide is roughly like 47,000 times 40,000 pixel. Its raw data is like 0.4 gigabytes, and if we store it on disk, it's like 300 megabytes after compression. The way we handle this type of data is we create pyramid structures. So we rescale the images at different magnification levels. For example, if you scan the image at 20x, you can try to rescale then maybe right up to like 10x, 5x, and 2.x. So we can store different zoom level of the images in the same file. So when we try to take a look at the image, we don't need to open the 20x immediately. We can just start from a very high level of zooming. So this consumes less memory and allow us to work with this type of images. So, okay. Give me a minute. Okay. So to work with this type of data sets, we propose a data structure called host-like image data. So WSI data, it's actually built on top of spatial data and is not very different from spatial data. We didn't do any modification of the original spatial data. It's merely a records that combines host-like image readers and the spatial data structures to store the corresponding information. So the purpose of creating such data structure is because many of the file formats in the host-like image words are quite proprietary. We don't know the underlying structures, and there are limited numbers of readers that we can open it, and we cannot really convert this image format to, let's say, the open format that's compatible with spatial data. And another benefit of using WSI data compared to other host-like image software you can find on the market is that this allows you to very easily integrate with the Python data science ecosystem. If you're using data science software like NumPy, Pandas, or other data structures, you can easily convert from whatever part of the spatial data to this kind of data types. And we also provide user with a very nice interface to run a lot of deep learning applications. For example, if you want to train a classifier, if you want to train a segmentation models, or even build up the foundation models yourself to run some self-supervised learning, it's all possible. So the lazy slide is built on top of this data structure. And this also allows us to integrate with everything that's happening in the SC-verse. So you can also try to analyze the host-like image dataset as if you are working with single-cell data. So let's take a quick look at this data format. So here I use a GTEX slide as an example. So when you want to open host-like images, all you need to do is to import this OpenWSI API from the WSI data package. You just call it OpenWSI and point it to the file. And you will see that if you print or print this WSI data object, you will see these very nice representations. So you will see like a image, a thumbnail on the left side, and the relevant information of the slide on the right side. I think this is very good for users, so we can basically easily access necessary information with this kind of rich object representation. So how do we process a host-like images? As we've discussed before, the host-like image, they are very big. It's a very huge images. So the idea is to try to break apart these images into smaller pieces so we can actually handle in our computer. The first part is to do tissue segmentations. We identify the tissue region from the whole images. So because the background are usually contains very little information to run the analysis, so we will only focus on the part where there is tissues. And then we would try to tile this tissue. We try to break the tissue images into very tiny pieces. So for example, here we tile the tissue into a 512 times 512 tile size. And this allows us to also run these tiles through a vision model, because as you know, if we want to embed images into some features space, you will need to have square line images and they must have the same sizes. So with these three steps combined allows you to already created a single cell-like data, which is each row is a tile and each column is a features. So the feature is usually created by either a vision model or a foundation model that's available on the market. And in Lazy Slide, after you open your WSI, you can easily create such data with 3 lines of code, as we can show in the top right. So let's look at these features. These features are seen by the model and usually capture the morphological parts of the slide, the morphological features. So these features are usually stored in an annotated data object that you are familiar with if you know about single cell, and it can be easily processed by ScanPy. For example, if we run some clustering on these morphological features, it can review us with different morphological archetypes. For example, here we run some Leiden clustering and get 4 clusters. You can see on the feature space that they are quite different. Like in each archetype, they are defined by different parts of the features. And if we try to project this the morphological archetypes back to the tissue images, you can find that it actually capture different like those parts, different spatial domain of the tissues, and they're actually visual distinct to us. For example, the green part is actually corresponding to the lymphocyte region of these tissues. And this basically allow us to perform spatial clustering or spatial domain analysis that's very popular in the spatial omics, but here we do it on the whole slide imaging parts. And so a further things we can do is to use text to perform analysis. So with this multi-model foundation model, for example, here I show an example of conch. So this vision language foundation model works like this. So you try to encode the images and the text into the same embedding space. And then you can try to calculate the distance between the text and the images because they're in the same embedding space. So you can easily calculate distance with, for example, cosine distance to see how close the text to the images is. And this allows you to perform tasks like zero-shot clustering or other prediction tasks. So here we show an example of how we use 4 tags: mucosa, submucosa, muscularius, and lymphocyte to query these tissue images. As you can see, because this is cosine distance and they're not normalized, So if the value is higher, it means the text and the images, they're more similar to each other's. So as you can appreciate, for example, the lymphocyte is the most obvious one. If the text and the lymphocyte region, they're mostly close to each other because you can see those red signal light up in the corresponding regions. So this basically allows you to analyze the host slide images with some text terms. So these are just some general structural terms here, but you can also use some pathological terms like tumors or like fibrosis to query your tissue to find the regions that have these characteristics, for example. And apart from using text, we can also try to integrate with, for example, RNA-seq with our host line image datasets. So here I use a very tiny dataset I constructed from the GTEx. So we have both healthy artery and calcified arteries. And by using this text image model that we discussed before, we can actually derive a calcification score to score all our slides. So this calcification score are merely just, I use this text, this exact term, calcification, to query the images. And then we can use this, their distance to score, to like provide a score to represent different slides. So as you can see, the calcification slides are usually higher than healthy slides in this bar chart. And we can further try to, like with this score, we can try to integrate the RNA-seq dataset with our morphological features. So if you can appreciate, we can see from this UMAP space on the left side, that's the host line image, because we see from the images, as you can see, there are some dark parts of the calcification in the calcification artery, and it's mostly red space in a healthy slide. So with the vision feature can actually capture more variance between the two groups compared to the genomic space. And this can also be reviewed by MOFA, that there are more variants in this type of data set on the WSI, on the host line image modality compared to the genomic space. And with the RNA linker in lazy slide, this can help you to use scoring. For example, a scoring we derive from text image similarity analysis to supervise the RNA-seq analysis. For example, here we run some pathway analysis with this kind of integrations. So as you can see, if we combine the information from the host slide images, we can actually review a much more specific pathway. For example, the cellular response to interleukin-18, this kind of specific pathway to the calcification process, compared to solely the RNA, which are quite general pathways. So I hope you can appreciate that how host site image can add a value to your genomic analysis. And apart from all this multi-model integration, we can also perform, for example, segmentation analysis on our data sets. So on the left side, we run semantic analysis, semantic segmentation to segment different regions of the slide. So here it's a mouse lung tissue, and we try to segment the airway, vessel, and the tumor from our tissue. So there's unfortunately there is no like such thing called foundation segmentation model exists on the market yet. but you can also train your own model to perform this kind of semantic analysis. But if you want to do cell segmentation on the host line image tissue, you can also use lazy slide with different models. So because there are a lot of cell segmentation models on the market, and we provide you with a very nice interface to run it across the host slide and help you merge the whole result into and project it onto the host slide. So we also have a model, for example, like CellVIT, that allows you to not only do segmentation, but also perform classification of cell types while you're doing segmentation. And if you can see is on the lower right side, this is a lung cancer tissue, and most of the cell type they're classified as neoplastic, and some of the cell type they're either epithelial or inflammatory. So I would say on most of the cancer tissue, these models, its classification part perform pretty well. And if you want to use this model on some healthy tissue, it may not perform as expected because it's trained on the cancer tissue. But the segmentation part of the tissue, it's of this model, it's working also pretty well. So other things we can do with host line images is I've introduced a bit before. It's called the zero-shot classification. So if you're not familiar with the zero-shot learning, this basically means with the pre-trained models, you don't need to train anything for this model to acquire a decent result using this model. For example, the text image foundation model, they're pre-trained by closing the gap between the text and images. But without any training, we can use it for classification tasks. This is kind of usage are called zero-shot learning. So here we shows an example to use two text words, two terms, healthy and calcify, to curate our tissue to classify whether it's a healthy artery slide or a calcified artery slide. So on the right-hand side, here we use a confusion matrix to represent the accuracy or the performance of the zero-shot classification. As you can see, I personally consider, given that we didn't train anything of this model to this specific test, this performance is pretty good because calcification, it's a chronic disease, so there will be some intermediate state, like the transition between healthy and calcification. The model not only just like divide this two category into two extreme part, but also capture some intermediate state. We also have another example. We try to do zero-shot organ classification. So here we have organs slides such as brain, lung, liver, and with the zero-shot classifications, you can see that the model kind of make all of the prediction correct, but some of them the probability are a little bit low because at the morphological space, they could be look quite closely to other tissue types. So this also depends on whether you can tell the differences at the morphological part. So we can also use what we've achieved, what we've acquired from the text image similarity to perform the zero-shot segmentation. As I've discussed before, we don't have existing model as like foundation segmentation model. But one thing we can do is to use the existing powerful segmentation models such as the SAM, the segment anything model from Meta, and use our text image query result as a prompt to try to perform the zero-shot segmentations. For example, here I show we try to threshold the lymphocyte image similarity, and we use these terms as either like the positive part and the negative part to perform a lymphoid segmentation using SAM. So, however, this approaches its performance is largely depends on how good the text image similarity is. It may not be very stable across slides or across different data sets. And here it's a list of supportive foundation models in lazy slides, and we are also extending the list as we evolve or with more foundation models appears on the markets. You also feel free to request different models from us in the GitHub issues if you find something that are useful, and we should integrate it. So for lazy slide, you can also find very nice online materials in our tutorial or the documentation websites. We have different tutorials to cover different aspects of the analysis. And we are also adding more tutorials to the documentation. For example, we are working on a survival analysis example using the TCGA datasets. And this will be happen probably like release in the next month or so. And there's already some projects using LazySci, for example, this spatial atlas of human anatomy, SAHAT. So it's an international joint effort trying to profile the spatial omics of the human tissues. And I'm very glad that we're using LazySlide to profile the whole slide image and combine with some spatial omics data in this dataset and in this project. And if you want, you can also try to dig out some useful information from the whole slide images and use LazySlide in your outstanding projects. And in the end, I would like to encourage everyone in this meeting to take a look at our packages. So these are the two GitHub repo. You can find our packages, both the LazySlide and the WSI data. And I would really appreciate if you can give us a stars, as this will help us get more public attentions and help us keep developing these packages. So we are part of the SCVerse ecosystem by now, and we have currently we only have a preprint on the bioarchive, and the manuscript of the LazySci are still in revision or so, and we will see if we can make it. And thank you for inviting me, and I hope you like LazySlide.